The overall goal of this protocol is to use cellular vehicles containing gold nanorods as an innovative strategy to deliver plasmonic particles into tumors.
Our protocol can help answer key questions on the development of photoacoustic and photothermal approaches to image and treat cancer by the use of exogenous contrast agents.
This method rests on the tropism of tumor-associated macrophages to deliver plasmonic particles to tumors. The idea is to isolate these cells from a patient, load them with plasmonic particles in vitro, and then inject them back into their host with the intent to use them as Trojan horses.
One of the advantages of this approach is the possibility of gaining more control over the optical and biochemical stability of the particles, because their biological interface is fully constructed in vitro.
To begin, purify 6 milliliters of centrimonium-capped gold nanorods at a gold concentration of 450 micromolar using 2 centrifugations, followed by decanting and washing in 500 micromolar aqueous centrimonium bromide. Then, add 1.5 milliliters of 100 millimolar acetate buffer with centrimonium bromide and polysorbate 20 into the vessel containing the gold particle pellet. Then, add 7.5 microliters of 10 millimolar alpha-methoxy omega mercaptopolyethylene glycol in water and leave it to react for 30 minutes at 37 degrees Celsius. Next, add 7.5 microliters of 100 millimolar mercapto-undecyl trimethylammonium bromide in dimethyl sulfoxide. Then, let the reaction rest for 24 hours at 37 degrees Celsius. After 24